Just yesterday, this gentleman died. This is the Los Angeles Times reporting his death. Renato Dulbecco, 1975 Nobel Prize winner. We have mentioned him before. And of course, here in the Times, it says he developed a method for measuring the quantity of virus in cells in tissue culture, a finding that greatly facilitated the study of such viruses. So now, having been in this course, if you ever read this, you would immediately know that he developed which assay? The plaque assay. And he was the first to do it for animal viruses. He also was important for figuring out how viruses cause cancer. And that's the reason he got the Nobel Prize. So we'll be talking about him a little bit more later on in this course. 97, not bad, right? Be nice to make it. All right, so far in this course, we talk an awful lot about mRNA. Like every virus's goal is to get to mRNA. And so today, we're going to figure out what exactly you do with mRNA. We're going to talk about translation, making protein from mRNA. So here's been the goal of everything just about we've talked about for all these different viruses, getting to mRNA. And as you well know, mRNA is decoded, it's done in the cytoplasm, and it is the result of that decoding is we make proteins. So today we're going to talk about how the decoding works. A lot of this should be familiar to you. If you took biology, you learn all about translation. And then next time we're going to talk about how we take the genomes and the proteins and make new particles. So a lot of this will be review, so we'll go through it rather quickly. If you haven't learned about it and it's too fast, you should either replay the lectures or at a slow speed or check out the textbook because a lot of this is in there. So mRNAs have a very typical structure in eukaryotic cells. Here's a generic mRNA. The 5' end, there's a cap structure. We've talked about that a bit already. The cap is an unusual base linkage. As you know, in RNA and DNA, bases are linked in a 5' to 3' chemical direction. And a cap is nothing more than a 5' to 5' linkage between a guanosine and the next base of the RNA. So that's what a cap structure is. It's typically methylated on one or more bases. You can see here on the 7 position, there's a methyl group. Caps are on most eukaryotic RNAs except some organelle, like mitochondrial mRNAs. Some viral mRNAs don't have caps, we talked about before. It is important for a variety of functions in transport, processing, but for our purposes, it's important for translation, as we'll see. There is usually 5' and 3' non-coding regions on the messenger RNA and a 3' poly A tail. And in the middle is the open reading frame that encodes the protein. It begins with an AUG, but not always. Most of the time AUG. And then there's a stop codon. There are 3 different stop codons, of course. And here's again a look at this typical mRNA. The 5' untranslated region varies in length. It can be pretty short, typically 50 to 70 nucleotides, but there are examples of very long 5' UTRs. It often has secondary structure, which you know about already, base paired RNAs. And the amount of secondary structure in the 5' UTR can affect the passage of ribosomes. So the efficiency of translation of a message will be regulated by the amount of RNA secondary structure up here in the 5' end. The 3' end is also important for regulating translation efficiency, in particular initiation. We'll talk just briefly about that. mRNA stability determinants also there as well. And finally, the poly A tail is also needed for efficient translation of mRNAs. The translational machinery is pretty complicated, and remember, this is something that viruses don't encode. Ribosomes in eukaryotes, they are 80S. They sediment at 80S in sucrose gradients. They consist of 2 subunits, the 40 and the 60S, and they're made up of protein and RNA. And in fact, you can take the protein away and leave just the RNA, and that has catalytic activity. That can make peptide bonds, which lends more credence to the idea that there was once an RNA world, and it was maybe the ribosome that were catalytic at that time. tRNAs are another important part of the translational machinery. I'm sure these are familiar to you. These are short RNAs that are highly structured. There's a lot of base pairing in them. They contain the anti-codon sequence, which matches with a complementary sequence on mRNA. And that's, of course, how amino acids are specified. The amino acids are attached up here at this 3' end. And they have structures, and they're quite complex, as you can see here. In addition to ribosomes and tRNAs, there are also many other proteins that are involved in translation. And they're given very specific names. We're going to encounter these throughout this lecture. They're all beginning with a little e for eukaryotic, and then they're either initiation factors or elongation factors or termination factors. They used to be called factors before it was known that in fact they were proteins, so I call them proteins now because factor is just a very vague term, but the original abbreviations stick, and we'll talk about some of these today. So I first want to start by considering these two general mechanisms of initiation. So translation has to begin before it can continue, and the beginning part is called initiation. There are two general ways that initiation happens, and you probably learned about the first one in another course. It's called 5' dependent initiation. Most mRNAs are translated by this mechanism. What happens here, and we'll look at this in a moment, the initiation complex binds to the 5' end of the mRNA. It finds the AUG, or the initiation codon, and then begins to make protein. Another mechanism discovered in viruses and now found to operate in cells you may not be familiar with is called internal ribosome entry. And here the ribosome binds internally, not at the 5' end as for the other mechanism. So let's look at 5' dependent briefly, just to review it so we're all on the same level. In this mechanism, the ribosomes are brought to the 5' end. So here's the goal in the end here, our messenger RNA with a 5' cap. The cap is going to play a crucial role in getting the ribosome to the message. So in the cell, we have 3 subunits, and here's the 40S subunit. It's typically complexed with a couple of initiation proteins. You can see EIF3 and 1A. A very important step is the binding of the ternary complex. It's called ternary because there are 3 components: molecule of GTP, EIF2, and the tRNA MET. Actually, the GTP EIF2 is one component, EIF1 is the third. That is the initiator because it contains a methionine amino acid, a charged tRNA with methionine on it. That binds to the 40S subunit in the absence of any RNA. It then associates with further proteins, including this red one, EIF4G, and the green one, EIF4A. We'll talk about what those do. And then finally, this complex binds to the mRNA. And the way it binds is that the cap at the 5' end is bound specifically by this protein called EIF4E, the cap binding protein. And then 4E, in turn, binds EIF4G, which is the red proteins. These are terrible names, unfortunately. But mechanistically, these proteins are unique. Cap binding, and then you can see the tethering of the whole ribosomal subunit comes via 4G. So that's how this 40S subunit gets to the 5' end by this protein-protein cap interaction. And let's take a look at this EIF4G a little bit. This is a very large protein, about 220 or 250,000 Daltons, pretty big. Largely discovered because it is modified in virus-infected cells. Discovered a number of years ago because it was modified, as you'll see later. As I said, it's this red protein that links the cap-binding protein with the 40S subunit. It doesn't bind the 40S directly, but through another protein called EIF3. And it's got a number of domains in it. You can see it has an EIF4E binding domain. Of course, that's this little part here that binds 4E. It has an EIF3 binding domain, a 4A binding domain, which is this protein here at the end. And you'll see later it has a binding domain for poly-A binding protein. We'll see what that does in a moment. Now, this protein is modified in cells infected with some viruses. That's what the red arrow means, and we'll talk about that later on today. So we've got so far an initiation complex on our mRNA. That's from 2 slides ago. And then the next thing, it has to find that first initiation codon, which, as I said, is mostly a AUG codon, but there are exceptions, as you'll see later. And we think that the complex either moves down the RNA or the RNA threads through the complex. We're not sure which one, but somehow this initiator gets to this initiation complex. Whether it can be the first AUG, but not always. It could be a second or a third. There are sequences around the AUG that control its selection as an initiator. And an initiator means you begin protein synthesis. So the movement of this complex, or the RNA, requires energy in the form of ATP hydrolysis. That's because this protein EIF4A is actually an RNA helicase. It unwinds RNA structures. So here's an RNA duplex, a stem loop structure, and it's in the way of the ribosome complex moving. 4A unwinds it, and that unwinding needs energy. And the more structures you have, the more you need this helicase to unwind these structures. Otherwise, the RNA, the ribosome, cannot move past them. So here we show the complex moving down. When it reaches the AUG, a lot of things happen. The GTP here is hydrolyzed, and EIF2 is released, along with a lot of the other proteins that got the complex here. And most importantly, the tRNA met remains, and the big subunit comes in. We call this large subunit joining. That's triggered when the AUG is found. And now we have an initiation ribosome ready to start making proteins. It's got the met in the P site here of the ribosome, and it's ready to go. There is an interesting role of the poly-A tail in translation, which is illustrated on this slide. We know that you need poly-A for efficient translation of proteins, and we think it's because the RNA actually forms this kind of circle structure. And the way this happens is that poly-A binding protein is a cellular protein that binds poly-A sequences in cells. It binds to the 3' poly-A, and it also binds to EIF4G. Remember, there's there's a poly-A binding protein binding site on EIF4G, and this binding and interaction circularizes the mRNA, and that's required to get efficient translation. So you can do a number of experiments to prove this. You can take off the poly-A, you can mutate these interaction sites, and you can show that all decreases translation efficiency. One of the ideas is that this allows ribosomes to recycle around very efficiently on the mRNA in which they're translated. We don't know if that's correct or not, but that's one idea. Okay, now in virus-infected cells, this is a basic mechanism that we've just talked about, which happens in uninfected cells and in most virus-infected cells. But there are modifications to the translation system, and that's what mainly we want to talk about today. And you can look at these in two ways. First, the cell is trying to get rid of the virus, so it makes alterations in the translation apparatus. We'll see how that happens in a bit. Now, this may sound odd to you that the cell would shut down translation or modify it in some way, but it's sort of a sacrificial move. The cell is willing to die to prevent the spread of infection. So the cells can modify their translational program when a virus comes in, and viruses also modify the translation of the cell because they would like it all dedicated to their mRNAs. They don't want to compete with cellular mRNAs. So there are many mechanisms at play. We'll talk about the translationally specific mechanisms today. Now, there is one virus-specific mechanism we talk about first. It's called ribosome shunting. And this is something viruses do to get around limitations placed on them by the host. And it's illustrated in this experiment. What we have here is a mRNA that we have created in the lab. And it's hooked up. It's got a lot of secondary structure here in the 5' UTR. It's capped. And there's an open reading frame which encodes for two proteins that we can easily detect on a gel. So we put this RNA, say, into an in vitro translation system. We translate it, and we run the products on a gel. And you've used this wild type sequence. You see you get two proteins. So the ribosomes can deal with this amount of secondary structure, and you get a nice amount of protein. Now we put a stem loop structure, this red one, in two different places, near the 3' end or near the 5' end of the 5' non-coding region. And this has a very high specific energy, minus 80 kcals per mole. In other words, it takes a lot of energy to melt it. And this is about the limit. ribosomes typically can't get by this. Even with a nice helicase EIF1A, they can't get by this. So you can effectively shut off translation by putting this in. So you can see here, when you put the insertion at the 5' end, it blocks translation. So the ribosomes can't scan by it, or they can't move by it. But look, if you put it near the 3' end, right near the AUG, that doesn't have any effect on translatability. So this was one experiment that suggested that at some point the ribosomes stop moving and somehow they bypass any structures that you've put in, like this one here. And in fact, that's what shunting is. The ribosomes scan for a little while and then somehow they are moving away from all the tertiary structure and finding the AUG. And here's a model for that. We've got our messenger RNA structured, ribosomes bound to the cap. That's how the ribosome gets to the message, starts to move along, and then at some point it shunts. And we're illustrating this as a way that the ribosome is bypassing all this structure. It goes from top to top, treetop to treetop maybe. Instead of having to go up and down each tree, it's leaping in some way. So that's the metaphor. We don't actually know how this works, but you have to imagine that this RNA is quite knotted up, and maybe the ribosome can just slide on the top of these stem loop structures. And that's why putting one down here, putting a stable stem loop down near the AUG doesn't have any effect on translation, because the ribosome is just passing over it. So that is shunting. It's seen in mRNAs of a couple of different viruses as shown here. Why is it important? Well, many of the responses of a cell to inhibit translation when a virus infects them is to reduce the ability of the cap binding protein complexes to be recruited to viral messenger RNAs. So the cap binding complex, that whole complex of proteins, including the helicase, is very important when there's a lot of secondary structure on the messenger RNA. But here we don't care about the secondary structure anymore, because the ribosomes are shunting over it. So the idea is that this decreases the dependence of any particular mRNA in having a lot of the cap binding protein complex, which can be reduced by the cell. So it's in effect a response to cells shutting off translation. All right, so that's one mechanism, ribosome shunting. This is a unique virus-specific mechanism. Here's one that was first discovered in viruses, but is now known not to be unique to viruses. It's called internal initiation. And this was first discovered when the genome of poliovirus was sequenced way back in the 80s, when the nucleotide sequence was determined. This was the first time it was figured out for this virus. It was found that the initiation codon, which begins this very long polyprotein, starts about 740 bases from the 5' end. And furthermore, this supposedly non-coding region was full of AUG codons, had 8 of them, and that was unprecedented, and no one could really understand how a ribosome could bind the 5' end and scan through all of these. Finally, the 5' end of polio RNA doesn't have a cap. It's got a little protein on it. Remember, that's the protein that you need for priming of RNA synthesis. So no one understood how a ribosome could bind to the 5' end of this message and scan to the right AUG. So it turns out that the ribosomes don't bind to the 5' end. They bind internally, very near to the AUG, and that's called internal initiation. And this was the experiment done to prove that. So here, what we have done was to create 2 different artificial mRNAs. We do this by recombinant DNA technology. We make these mRNAs in a plasmid, and then we put the plasmid in cells, and the plasmids make mRNA. So here's one on the left. It's a capped mRNA, has poly A, and there are 2 open reading frames encoding 2 different proteins. Now this was done in the 80s, so we're using thymidine kinase and a protein called CAT. If you were doing this today, you would use 2 different kinds of luciferase or a fluorescent protein. The point is you can assay for these in a very easy way. So you have this mRNA with 2 different open reading frames, and then in between there's a little spacer sequence. In this case, it's just a random nucleotide sequence. Here, we've taken the 5' non-coding region of poliovirus, that long 700 and so base sequence, we've put it into here. because we think it might be allowing ribosomes to enter internally, so we're going to test that. Now, if you translate this RNA on the left in cells, you make quite a bit of this first protein, TK. You make a very small amount of the cat protein. So what's happening is the ribosomes will translate this TK, and then after reaching the termination codon at the end of it, they fall off. Most of them fall off. This is typical for eukaryotic cells. A few do scan through and translate the second. You get a little bit of that second protein. If you do the same experiment with an RNA that now has that 5' non-coding region from polio, you get a lot of both kinds of proteins. So apparently the ribosomes can initiate internally, and you get much more cat synthesis here than here. Now that suggests very strongly, but doesn't prove that in fact this is allowing internal ribosome entry. So there are two more experiments I'm going to tell you that prove that. The first is on the bottom. You do the same experiment, except now you do it in a poliovirus infected cell. And as you will see later on today, what polio does is to shut off 5' end dependent translation initiation. So that first mechanism that we talked about, where ribosomes bind the 5' end and scan in, that is shut off by polio. That's what the little red circle with the line in it means here. It means ribosomes can no longer bind to the 5' end. So that's a good experiment because it rules out the possibility that all this sequence does here is to allow ribosomes to keep riding through from the first open reading frame. So if you put this RNA into polio-infected cells, neither protein is made. You can't get any of the first one made because there's no 5' end initiation. You can't get any of the second one made because that's made by scanning or read-through. However, if you put this RNA into cells, you get only the second protein translated because, again, the first one is inhibited because 5' end translation is shut down by the virus. And the second works because the ribosomes apparently can bind internally. So that was good proof for internal initiation. There were still some skeptics, and they said, I want you to, if this is true, this means that an internal initiation mechanism does not require a free 5' end. So they said, make an RNA without a free 5' end. So it took a long time to do that because it was technically difficult. But in the end, someone made circular RNAs, which you can do by ligating the ends together. And they made one RNA here with an internal ribosome entry sequence from polio. And then there's an open reading frame. It's in yellow. So if you translate these, you would theoretically get that protein. We have two different circular RNAs, one with an internal sequence. That's the 5' UTR from polio. And one without. And you translate these in vitro, you get protein from this one, but not from the one without the polio 5' UTR. So it shows that ribosomes can bind internally here, but not without this sequence. So this sequence was then called an internal ribosome entry site, or IRIS. So now this is the second mechanism of translation initiation. And irises have a very specific look to them. They are highly structured RNA molecules. So here is the iris of poliovirus at the 5' end. It's that sequence of bases, which I first showed you as just a line of bases. It actually folds extensively on itself. This is the iris of hepatitis C virus, a different family of viruses. This is a different picornavirus right here. And this is an insect picornavirus, cricket paralysis virus. These have since been found in many, many other viruses, and they seem to work by allowing the ribosome to bind internally. And in fact, for this iris, we know that EIF4G, that red linker protein, can bind directly to this structured RNA. So EIF4G binds, and then the ribosome can bind to it. You don't need caps at all. That's why these work. The hepatitis C virus iris is very interesting because it will bind the 40S subunit directly without any other protein. So this has evolved over time such that 40S will bind this highly structured RNA. So the 40S subunit binds. You don't need a cap. You don't need a 5' cap binding protein. And then the EIF3 comes in and the rest of initiation occurs. So it's a very simplistic iris in terms of how they work. So these two mechanisms of initiation are summarized here. So here is 5' end dependent, which we started talking about, where the cap recruits the initiation complex. And this is internal ribosome entry, either with a picornavirus iris or the hep C iris. So for the picornas, we think that 4G binds the RNA directly, and that brings in the 40S subunit. You don't need a cap, and these RNAs are not capped, in fact. And the hep C iris, the 40S subunit can bind the structured RNA directly. You don't need an intermediary like 4G. Now, you notice here that the N-terminus of 4G is broken, and that will become obvious to you later. This is done by viruses in infected cells. They can cleave EIF 4G and inactivate it as far as its function in cap binding goes. But the cleaved 4G, which is what you find in, say, a picornavirus infected cell, is still fully competent to interact with the iris and stimulate internal initiation. That's important because the virus can still use this protein and translate, but the cell can't. So there are many viruses that now have been shown to have irises, shown on the left here, just to give you an idea. Even retroviruses have irises. And furthermore, cellular mRNAs have been shown to have irises as well. And these are thought to be needed because in cells, when you go through mitosis, this 5' end-dependent translation is actually shut off. Cells specifically do this during mitosis. But there are some proteins you need made during mitosis, and those are typically made using an iris because there's no shutoff of iris-dependent or internal initiation type translation. Okay, so we have so far talked about initiation, and we've got this initiation complex either at the 5' end or internally. We're now at the initiation codon, and I'm showing it here as an AUG. This is where we left off before we diverged and talked about virus-specific mechanisms. So we have our mRNA, and we have our fully assembled ribosome with the met tRNA in the P site. And this could have been brought here by scanning, or it could be brought here by internal initiation. Doesn't really matter. What happens next? Well, you know, the next step, of course, is the insertion of the next amino acid by another tRNA here. But before we talk about that, I want to tell you how some viruses can actually go about initiating without a methionine tRNA. Now, so far I've shown you initiation where this ternary complex, the EIF2 complexed with a tRNA with methionine, is absolutely essential for translation. But there are a couple of virus-specified cases where it's not. And this is very interesting. This is called methionine-independent initiation. So there are two examples shown here. And there is one here for a picornavirus of crickets, cricket paralysis virus. It's basically polio of crickets. And this has a structure in the RNA that can fold to mimic a met tRNA. So what happens is this RNA can initiate translation without any met tRNA. It uses its own RNA to fold and base pair with this initiation codon, which you can see is not an AUG. Here it's a proline, CCU. And then the next amino acid that goes in, or the first amino acid that goes in, would be the alanine specified by the next codon. So this virus gets around any need for this ternary complex, which can be limiting in virus infection because cells, as you will see, can shut down its availability. This leads to a side consideration, which is if you're looking for a gene, let's say you sequence a new organism and you're looking to see how many proteins it encodes. If you only look for AUG initiating open reading frames, you might miss some of these because these don't begin with AUG. And there are a number of these in mammalian genomes, for example, plant genomes. We suspect that they may, that is, open reading frames that are quite long that don't have an AUG, we suspect they may be translated by these sorts of mechanisms as well. The second one is shown here. This is actually a plant viral RNA. These are both positive strand RNA viruses. And this one is very interesting. The 3' end of this viral RNA can fold into a tRNA-like structure. You can see it right here. And it is actually so much like a tRNA that the cell sees it and puts an amino acid on it. It charges it, and in this case it's a valine. And that's done, of course, by tRNA synthetases. This tRNA-like structure sits into the P site. In other words, it's a substitute for the MET initiation tRNA. And so again, the initiation codon is not an AUG, it's a valine, a GUG in this case, and then the second amino acid would go in here. So just two ways to show you that methionine is not always the way to initiate protein translation. Now this is an expansion on this interesting poliovirus of cricket paralysis virus. It is an RNA virus much like polio. It's a positive strand RNA, has a VPG at the 5' end, it's polyadenylated. But if you remember, polio RNA encoded one long protein. This virus has its open reading frame broken into two pieces with an iris in between. So not only is there an iris up here at the 5' end, just like there is with polio, but there's one in between these two open reading frames, and that is the iris shown here, and it's the one where the RNA actually folds and mimics a initiating tRNA. And so you may ask, why does this have two open reading frames, but polio does not? Well, I can't answer that. It just seems to work. But again, this is also interesting because this iris, not only does it mimic a tRNA, initiation tRNA, but this iris can bind to 40S subunits without any other proteins present. So this actually brings us to a more general consideration in translation strategies, and that is reflected in this slide, which shows a fundamental difference between mRNA translation in eukaryotes and compared with bacteria and archaea. So in bacteria and archaea, as you may know, a single mRNA can encode multiple proteins. And here we have 3 different proteins. And each of those proteins has an initiator AUG and a stop codon. And the ribosomes can identify each of these initiation codons because they're surrounded by the so-called Schindel-Garneau sequence, which is a sequence in the message that's complementary to some ribosomal RNA sequence. So in a way, these bacterial and archaeal messages have sort of like internal initiation sequences. Eukaryotic mRNAs, by contrast, are largely monocistronic. It means that they encode one protein, shown up here. And that's because except for irises, which I've told you about, there are no sequences that allow ribosomes to bind internally. So once a ribosome makes a protein, it hits a stop codon and it falls off of the messenger RNA. And if you had set up a eukaryotic mRNA to look like this, you'd get mostly the first protein made, a little bit of the second one, because most of the ribosomes would fall off, and none of the third. Unless, of course, you put irises in between each of these in a eukaryotic cell. But we don't actually see eukaryotic genes arrayed with irises in between them. So for the most part, they are functionally monocistronic. Now, viruses have to get around this, right? Because remember, some viral genomes encode a lot of proteins. How do you make many proteins from a single mRNA if it's a monocistronic mRNA? So that's what I want to talk about a little bit now, is how you would get around this limitation. And of course, you already know one answer. You could make a lot of messenger RNAs like VSV does. It makes 6 or 7 mRNAs to access all the coding regions. So let's talk about how that might work. So these are 6 different ways that you can access information in a viral RNA. We're going to just talk about a few of these today. We've talked about internal initiation. We're going to talk about polyproteins and leaky scanning. And then we'll look at suppression and ribosomal frame shifting. And these are all mechanisms found in a variety of virus families. So the first question is, you have a plus strand mRNA in a virus like polio or a picorna. It encodes a number of different proteins. It encodes 12 proteins. How do you get all those if the mRNA is monocistronic? Well, the picornas use this polyprotein synthesis, right? They make one long protein, and then they chop it up into smaller proteins using proteases. I think we've talked about this already. Here is the polyprotein translated from a picornaviral message. And that polyprotein has embedded in it 2 proteases. These are enzymes that cleave proteins And those can not only clip themselves out of the polyprotein, but they can also act on other polyproteins in trans. And there are two of them. They're called 2A and 3C. The names are not important. What is important is that these process the polyprotein, and you get all the individual polypeptides. And these blue and red arrows are all the processing sites for these viral proteases. And so you see among these are the capsid proteins, and then proteins involved in replication, like the RNA polymerase down here. So that is a picornaviral strategy. Below it is a flavivirus like Hep C, uses the same kind of strategy, has a plus strand RNA, makes a big protein. Simple to get all the proteins from it just by processing. Okay, then of course, you might say, can we use an iris to separate all these individual proteins? And the answer is usually not. The only example I know of is that insect picornavirus, where you have two open reading frames with an iris in between. That is the only example I know of splitting up proteins by an iris in a virus. Usually the irises are at the 5' end. Now, another way is to get many proteins from a single message is called leaky scanning. So now this is in a negative strand RNA virus. These, remember, like influenza or measles virus, these have, in the case of measles, they have single negative strand RNAs. So of course a negative strand RNA can't be translated when it gets into a cell. It has to be copied to a plus strand mRNA. And these viruses usually make multiple mRNAs from the long genome. And that's how they access all the individual protein coding information. They make individual mRNAs. But they even go further. Some of these viruses make multiple proteins from each mRNA. They maximize their coding potential. And this mechanism of leaky scanning is one way that you can do that. So we're looking at a very specific mRNA from a paramyxovirus, and it encodes these C proteins. But you can see here that you can make a lot of different proteins from this single mRNA. Now, this is a very typical message. It's capped, it's polyadenylated, but it can make 1, 2, 3, 4, 5, 6, 7, 8 different proteins from this one message. And I just want to talk about two of the mechanisms that give rise to this, because it shows you how the viruses can bend the rules. So we're looking at this very region here at the left part of the mRNA. It's blown up below here. And you can see that here's our cap. Now, if a ribosome bound to this cap, let's say, would start scanning, and the first place it starts to translate protein is actually not an AUG, it's an ACG at position 81. And that gives you this protein C'. This is an ACG, so it's not a particularly good initiator. So maybe 5% of the ribosomes will initiate there, but the other 95% will keep on moving. That's why we call it leaky scanning, because they scan, they initiate, but many of them keep moving downstream. The next codon that you could initiate at is an AUG at 104. And some ribosomes, in fact, do that. And that makes a different protein called P, in a different reading frame, so you can see it's a different color. But this AUG turns out not to be a great AUG. The surrounding sequences are not optimal. So again, many ribosomes bypass it. Some do initiate, but many keep going. And they reach then finally the AUG at 114, which is a great AUG. It's in a good context, and then all the ribosomes, the rest of the ribosomes initiate there. So we've now made 3 different proteins by this leaky scanning method. Now, you may be wondering about these two here. They're quite further downstream, 183 and 201. These are not made by scanning. These are made by ribosome shunting. So it turns out that some ribosomes actually jump from the 5' non-coding region all the way to these individual AUGs by the mechanism that we talked about previously. So at least 5 proteins here made by a combination of leaky scanning and shunting. And if you're interested, this one, the brown and the blue proteins are made by mRNA editing. You remember, you can have some non-templated mRNAs made in these viruses that add extra bases in the message that are not templated. So that happens 5 or 10% of the time, and those non-templated bases cause a shift in the open reading frame, so you get different proteins. So this is a really remarkable mRNA that you can make so many different proteins from it. And it just goes to show you can get around this monocistronic rule very easily. Another way is to change the termination of proteins. You'll see this is very common in viruses. Now, when you translate a protein, you initiate it at an initiation codon. Eventually, you reach a stop codon, and there are 3 of them, UAA, UAG, and UGA. And one of them is shown here. And what happens is these are specifically recognized by release factors, ERF1 and 3. They mimic, ERF1 mimics a tRNA in its structure, and it fits into this termination codon site, and it causes termination of protein synthesis and release of the polypeptide chain. That's why they're called release factors. Now and then, these stop codons are recognized by a tRNA that has an amino acid in it, and that is called suppression because we are suppressing the termination signal. And then translation can keep going, so you can make a longer protein. And this turns out to have a lot of importance for the life cycles of a number of viruses. Okay, so normally you terminate here, but sometimes 5 to 10% of the time you can get suppression and translation. So here are 2 different viral genomes where this happens. On the left is a retrovirus genome. Now, you may remember from last time that some retroviruses make an mRNA that encodes the left-hand part of the genome, the GAG and the POL proteins. And then there's also information coded to the right, the envelope glycoproteins. Those are made by a spliced message. So here's the message that encodes the GAG and the POL proteins. These are the structural proteins, the GAG, and this is the polymerase, the reverse transcriptase, the RNase H, the integrase. Now, this is the typical message. It's capped and polyadenylated. The ribosomes initiate. They translate the GAG protein, but there's a stop codon at the end of the GAG protein. That's shown right here, this UAG. So most of the time, the ribosomes stop, and you just have GAG protein made. And that's fine because you need a lot of GAG. It's a structural protein. But you have to have POL. You can't get away without having POL because this encodes reverse transcriptase that has to be packaged into the new particles. So what happens is there's a suppression event at this termination codon, so that 5 to 10% of the time the ribosomes keep translating because some tRNA is inserted here that has an amino acid attached to it. So that's one way that suppression is important. Now, why does this happen? We think this structure is important for regulating that event, that suppression event. This is not only a stem loop structure, but it's a pseudoknot because you have the unpaired bases here pairing with a sequence downstream. So that makes a pseudoknot structure. And we think this makes the ribosomes bump into it and pause. And when they do that, there's more likelihood to get a suppressor event. You can mutate this pseudoknot. If you eliminate these base pairs, you don't get any suppression anymore. So it lends credence to the idea that it's causing ribosomal pausing. On the right is the genomic RNA of an alpha virus like Synbis virus. And this is a plus strand RNA, which is in the particle. And as soon as this comes into a cell, it can be translated. And in fact, most of the RNAs are translated to make this P123 protein, which goes on to be processed further. But it's not the complete translation product. This protein ends here because there's a stop codon in this pink sequence in the RNA. Now, the proteins that are made from P123 are all important, but you need the 4th protein, which is the RNA polymerase, to be made. And again, that is made by suppression at a termination codon. You don't need a lot of it because it's an enzyme. It's catalytic. So this only happens about 5 to 10% of the time that the ribosomes are transiting, but it's enough to allow the virus to replicate. So those are 2 examples of how suppression is used to modulate synthesis. And it probably evolved this way because these are enzymes past the suppression site. You don't need a lot of them. Now, what happens when you get suppression? Sometimes it's simply a misreading. A normal tRNA with a normal amino acid charged linked to it will misread the termination codon, maybe because the ribosome is paused there and errors occur. But it's a normal tRNA. which is put in, so it puts in regular amino acid. But there are also specific suppressor tRNAs that recognize stop codons in cells. And these insert unusual amino acids like selenocysteine, which is put in for the terminator UGA. So just two different ways that you can get suppression, by a specific codon or by a mistake, a misreading. All right, so that is frame shifting. And one last mechanism that we will talk about in terms of getting the maximum of coding out of a genome, and this is frame shifting. And this happens also in retroviral genomes. Again, we're back to this retroviral mRNA that encodes a GAG pol protein. You remember we just talked about an example where there's a termination codon after the GAG protein, and that the GAG pol is made by suppression. There are some retroviruses where the two proteins are made separately by a frame shift event. So you have initiation at the 5' end. The GAG protein is made by translation starting from an AUG here. The ribosomes reach a termination codon. They stop making GAG. But then they back up a base. For some reason, they are moved back -1, so that shifts the reading frame. And then they start translating in the new frame. So they don't fall off. Somehow they stay on the message, because typically when you reach a terminator, the ribosome falls off. But in this case, they don't. They back up one, which, as you'll see, is probably a consequence of the nature of the RNA. And then they begin resuming. So you end up making two different proteins. You make actually a fusion protein because the ribosome is simply continuing to translate, doesn't actually terminate here. You make a fusion, very much like the fusion that you make when you suppress. And that is, the two proteins are then released by proteolysis. So that's called ribosomal frame shifting. I think you can see why it is, because again, the ribosomes back up, you change the reading frame. And that's illustrated here. So here we have one of these retroviral mRNAs being translated. There is a growing polypeptide chain attached to the tRNA in the P site. And then there's a new tRNA for the next codon put in the A site. And this kind of frame shifting often occurs at what are called slippery sequences. They are typically AU rich. And so if you back up just a base, you can maintain most of the base pairing. As you can see here, you have a U, A, UA, AU, 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 UA. But now we've gone back. The tRNA has slipped back one. And you can see you get pretty good base pairing, with the exception of this one. But now the ribosome is in a -1 frame. So as the ribosome moves forward, the next triplet is going to be a different tRNA than what it would have been. Of course, it would have been a termination codon right up here. So you no longer have a termination codon. You now actually incorporate an amino acid. The terminator UAG is now part of a new amino acid specified by AUA. Okay, so it slips back -1. There can actually be multiple ways of frame shifting in different multiples to get different reading frames. It's quite an interesting mechanism. It's another way to get more proteins for your genome. So all those get over this issue of monocystronic limitations in cells. They allow viruses to make many proteins. So now I want to turn to regulation of translation. And this, again, is from two perspectives. First, the cell is trying to regulate translation because it wants to limit virus infection. And the virus wants to shut off the host. It doesn't want any competition from host mRNAs. So let's take a look first back at the initiation step. There is a level of regulation that occurs here, which is quite pervasive. Initiation turns out to be the best way to regulate translation. It makes sense. It's the first step, which is not to say that there isn't regulation at elongation and termination, but most of the data comes from studies of initiation. Now, if you remember, when we reach the AUG codon, our 40S subunit, along with the ternary complex, you reach the AUG. The next step is going to be the addition of the 60S subunit. And when that happens, when the AUG is recognized, this GTP is hydrolyzed to GDP. And EIF2 is released along with it. And this will go on, of course, to initiate translation when the 60S comes in. But this molecule has to be recycled. this GDP has to be exchanged for GTP so that it can engage in another round of initiation. And the recycling is carried out by a protein called EIF2B, which is basically a guanine exchange factor. It binds GDP EIF2 and exchanges the GDP for a GTP molecule. All right, it actually exchanges the two. It doesn't phosphorylate GDP, exchanges the two. And now you have release of EIF2 GTP, and this can bind another tRNA and now becomes a ternary complex and can participate in another round. So every time you have a ribosome finding an AUG, you have this recycling event that's needed. So it turns out that this is a highly regulated step because there are protein kinases in cells that phosphorylate EIF2 and alter its recyclability. So here is a phosphorylation step occurring on EIF2. EIF2 gets phosphorylated on a very specific subunit. It's called the alpha subunit. And when there's a phosphate attached, it will still bind EIF2B, but it can't be released. All right, so normally EIF2B would exchange the GDP for GTP, but if it did that, EIF2 could not be released, so you would eventually sop up all the phosphorylated EIF2, and there would be no more initiation. So basically, by phosphorylating EIF2, you can turn off protein initiation. And cells do this when they're infected with viruses very early on in an attempt to stop virus multiplication. And there are 3 different protein kinases in cells that do this phosphorylation of EIF2. They're called PKR, PERK, and GCN2. And we're going to just focus on PKR, but I want you to know that there are other pathways that can lead to phosphorylation of this important protein. These kinases have double-stranded RNA binding domains. You'll see the reason they have that in a moment. And they have a kinase domain which allows them to phosphorylate EIF2. Right. Now, when viruses infect cells, there are very early responses initiated by the innate immune system. We'll talk a lot about this later on. Here's a virus infecting a cell. In this case, the genome is detected by the innate immune system as a foreign molecule. And that turns on the interferon system. This cell will now produce interferons. The interferons are secreted and bind to other cells and turn on the synthesis of a whole range of genes. They're called interferon-stimulated genes. And it's those genes that have antiviral properties. There are over 1,000 of them. One of those interferon-stimulated genes is PKR, the protein kinase that phosphorylates EIF2. So here we have the production of PKR, and this is made so that it can shut down translation and prevent the virus from spreading. Now, how does PKR work? As PKR is made, it is not active. It has to be activated. So if a virus infects a cell and interferon is made and PKR is eventually made, the PKR is not active. It has to be phosphorylated to be active. And the only way, there are 2 ways that it can be phosphorylated. There is actually a cell protein called PACT that will activate it. And there are, in fact, cellular reasons why you need PKR activated even in a non-infected cell. But we're not going to talk about that. What we're concerned about is down here, where inactive PKR is activated when it binds double-stranded RNA. And when it does that, these 2 PKRs line up on the double-stranded RNA and they phosphorylate each other. And now they're active. They can now go on and phosphorylate EIF2. Before that, they couldn't do anything to EIF2. So where does this double-stranded RNA come from? It comes from virus infections. You don't usually find double-stranded RNA in an uninfected cell. It's produced in the replication cycles of RNA viruses. The intermediates can be double-stranded. It can be secondary structure that is double-stranded. Even DNA viruses can make double-stranded RNAs. If you remember adenoviral DNA, it's a double-stranded DNA. There are promoters on both strands. So you make overlapping transcripts which can hybridize and make double-stranded RNA. So this is recognized as foreign, and it binds PKR and activates it. And then PKR can go on to phosphorylate EIF2 alpha. So only when the foreign RNA is present can this happen. So again, PKR is induced by virus infection. It's an interferon-stimulated gene. And the idea is to shut down translation. It also turns out that phosphorylation by PKR induces programmed cell death, which is another evasion strategy. And finally, the most important thing here, every virus that exists has some way of getting around the activity of PKR because it's so important. If viruses didn't get around it, they wouldn't exist. They'd be shut off out of existence. So every virus genome has some kind of PKR antagonist or some antagonist that interferes with this pathway. So I just want to show you two of them. And the first is in the genome of adenovirus. So adenoviruses, remember, are double-stranded DNA viruses. They can make double-stranded RNAs as a result of divergent transcription, as I just told you. And that double-stranded RNA can activate PKR because two PKR molecules can bind to it, and they can phosphorylate each other, and they will go on then to phosphorylate EIF2, and that would shut off translation. But adeno has evolved a small RNA called VA-RNA, which counteracts the activity of PKR. VA-RNA is double-stranded, so it binds PKR really well. But it's also very short. It can only bind one molecule of PKR. So PKR cannot be activated by binding VA-RNA because you need two PKRs binding on the same double-stranded molecule. So PKR is inactive, and it doesn't phosphorylate EIF2. So in fact, if you look in an adenovirus-infected cell in a very early hour or so of infection, you see phosphorylation of EIF2, translation starts to decline, but by then the virus starts to churn out VA-RNA, and that completely reverses the inhibition because it's binding up all the PKR. All right, so that's one example. A second one is encoded in the genome of vaccinia virus. So vaccinia encodes two proteins that antagonize PKR. They are called K3L and M156. Another two great names for proteins again. These structurally mimic EIF2 alpha. Here's the three-dimensional structure on the left of EIF2 alpha. And you can see these blue and green segments are very similar to the similar segments in these two proteins. These basically act as pseudo-substrates of PKR. So even if PKR is activated by phosphorylation, they will bind these two proteins. The virus makes a ton of these, and it will basically sop up all the PKR so it can never phosphorylate the real target, which is EIF2 alpha. So this is a different way of antagonizing the activity of PKR, but it works very well. And that's why these viruses can replicate. So we have learned for almost every virus that we've studied that there is some way to antagonize EIF2 alpha phosphorylation. This is an incredibly important target. And I just show you this. You don't need to know any of these, but you need to know that there are many ways that you can get around this phosphorylation. Look at the different viruses here and all the mechanisms of getting around EIF2 phosphorylation. So they're targets of double-stranded RNA. There are viral proteins that bind double-stranded RNA, like the VA RNA is an RNA, but there are also viral proteins that can bind and sequester it. There are some viral proteins that block the activity of PKR. There are pseudo-substrates, which we talked about for vaccinia virus, that block PKR phosphorylating its normal substrate. So it's clear from this kind of analysis that this is a really important step in regulation, and every virus wants to evolve around it. If they don't, they're in pretty much big trouble. All right, so that's one thing the cell does, tries to shut down translation. I've just told you one mechanism, EIF2 alpha phosphorylation. There are lots of others as well, but I think you get the point. Here's a second totally different way that a cell responds to infection. This is actually a stress response. If you stress cells in a number of ways, if you take away their serum, cells need serum to divide. They need the growth factors in the serum. If you heat shock them or UV irradiate them, they go into a stress response mode. And one of the things they do is to shut down translation, because if conditions are bad, there's no point in making proteins because they're going to go to waste. So you've got to halt protein synthesis. And one way of doing that, besides the EIF2 alpha phosphorylation, which is also a major player there, cells take all the mRNAs that are being translated and they shunt them into stress granules. And these are these yellow things here. If you look at a cytoplasm of an infected cell, you see it's full of these, not infected, a stressed cell. It'll be full of these stress granules, and within them are stalled mRNA complexes. So these are mRNAs that we're in the process of translating. The translation is shut down, and these are shunted into these stress granules. So these have a very specific structure. They're made of very specific cellular proteins. You can see some of them here, TIAR and G3BP. And the result is you shut down translation until such time that things are okay again. The stress goes away, the nutrients come back, then the stress granules are dissolved, and the mRNAs go back to being translational apparatuses. Now, if you're a virus infecting a cell, you're stressing it, and the cell makes, in response, stress granules. And that's not good for you because your viral mRNAs are never going to be translated. And so it turns out that many viruses reverse stress granule formation. And this has been best studied in a couple of viruses, West Nile and dengue, which are flaviviruses, and also poliovirus. If you infect cells with these viruses, you see very early after infection the assembly of stress granules in the cytoplasm. But then after a few hours, they go away. And they go away in the case of polio because one of the polio proteases cleaves a component of the stress granule, G3BP specifically. And it dissolves the stress granules, and the mRNAs that have been trapped there can come out and be translated, and newly synthesized messages will not be put into these stress granules because they're not there anymore. So this is a brand new discovery, just a couple years old. Probably has lots of implications beyond viral infection, but clearly a target of antagonism. All right, and finally, let's talk about what a virus does to the cell to inhibit translation, because as I've said, the virus wants the entire translational apparatus dedicated to it. And this is a mechanism that takes place in the coronavirus-infected cells, poliovirus, for example. And what these viruses do is cleave EIF4G, that red linker protein that links 4E to the ribosome. They cleave it right there near the N-terminus. And this whole complex, by the way, 4E, 4G, 4A, it's called EIF4F. So you're going to see some references to 4F in the remaining slides, and that's all that means, just another terminology. So 4G is cleaved, and it's cleaved by viral proteinases. In the case of polio, it's one of the two proteinases. And as I've said before, that is lethal for cap-dependent translation or 5' end-dependent translation. But the iris, or the internal initiation, doesn't care if this is cleaved because it's the rest of 4G that actually interacts with the internal ribosome entry sequence. So let's see what this means practically. So if you infect cells with polio, and you look at different hours after infection, it's a very rapid infection, and you look at the amount of protein synthesis, you see that the protein synthesis goes way down by 2.5 hours after infection. So the virus is shutting off cellular translation. And then it is replaced by viral translation here. And the cells die. And you can see this on this protein gel on the right. If you, at different times after infection, label cell proteins with S35 methionine, then you can run them out on a gel and detect them. You see at time 0 there are lots of cell proteins being made, but as time goes on, 1, 3, and 5 hours post-infection, you see the cell protein synthesis is gone. It's replaced all by viral proteins. So this is achieved by cleaving that EIF4G. So the virus turns off all cellular translation, and it's got the whole apparatus for itself. So again, why does this work? The cleavage of 4G blocks its ability to bind 4E, which is the cap binding protein. So capped messages no longer have a mechanism for recruiting the ribosome to the cap because this crucial linker, 4G, is now cleaved. So cellular translation is shut off because all the cell messages, for the most part, are capped. But the virus, and that's shown here, this cleavage right here destroys this nice assembly. But the virus needs only this cleaved portion of 4G to bind to the iris. So it makes perfect sense. The virus inhibits cell translation, but it has a mechanism of translating its own mRNAs that's independent of intact EIF4G, and that is internal ribosome entry. So that's just one way that this happens in cells. And there are many different strategies by other viruses, but the point is illustrated. Viruses need to have the apparatus devoted to their own translation. Finally, I want to end up on a different topic, which is increasingly becoming important in virology, and that is the regulation of translation by microRNAs. And these, of course, are small non-coding RNAs. They don't code protein. They're coded in most genomes. And we're beginning to see that they're in viral genomes as well as cellular genomes. And they're made by Pol2 or 3. They're made as precursors, and then they're processed by enzymes to 21 nucleotides. So this is the microRNA, 21 nucleotides. They're amazingly important for regulating gene expression. So our genome has over 1,000 microRNAs encoded in it. Some of them are tissue-specific, some of them are ubiquitous, and they control gene expression after transcription. So not at the level of transcription, but post-transcriptionally. They either degrade the mRNA or they inhibit translation, which is relevant to what we're talking about today. And most of them target the 3' non-coding region, but now about 30% of them seem to target elsewhere as well. So they can inhibit translation. And this is an example of a microRNA binding to a 3' non-coding region. Here's a messenger RNA, the end of the open reading frame and the poly A tail. They typically bind about 15 bases from the termination codon, and there are 2 areas of complementarity with a bulge in the middle. So this is 21 nucleotides in length. And how much complementarity you have here determines whether the message is degraded or if translation is inhibited. And this degradation requires a complex of other proteins, AGO, dicer, and TRBP. So again, this microRNA could either degrade this message, of course then it can't be translated, or it could inhibit translation on it. And we think that there are 2 ways that they regulate translation. These should now make perfect sense to you, having talked about mechanisms of initiation. So here we have the microRNA along with associated proteins, dicer and AGO, and so forth, binding. They're called microRNPs now, microribonucleoproteins. They're binding in the 3' UTR. We think these could actually inhibit initiation. We're not sure exactly at what step, but one idea is that they can interfere with either cap recognition, the recognition of the cap by 4E, or even by the joining of the 60S subunit. So they could affect at that distance. And now you may be wondering how this happens at a distance. Well, if these mRNAs are circularized by PABP, as I showed you earlier, this could have such an effect. These also are believed to inhibit elongation, that is the passage of ribosomes down the mRNA. This is not something we talked much about today, but some of the microRNAs seem to affect that step as well. So what's the relevance to viruses? Here's one example of a really interesting microRNA. This is called miR-122, so that's the terminology, microRNA, and they're given numbers. And this one is liver-specific. It's only found in the liver. And it is required for the replication of hepatitis C virus. So if you take away this microRNA, you can do this in cell culture. You can antagonize the production of specific microRNAs. Hep C replicates very poorly. So this is amazing. The virus has evolved to use or require a specific microRNA present in liver. So right now, there are in clinical trials drugs that bind to this microRNA and sequester it so the virus can't use it. And these drugs inhibit viral replication. So it's a brand new area of therapy. There are also microRNAs both in the cell and in the virus that target important genes. For example, we now know there are many cellular microRNAs that inhibit virus replication. And there are also viral microRNAs that inhibit immune responses in the host. So this is all new stuff because these are short RNAs that people used to think were junk. When you ran a gel of RNA, there was always some stuff at the bottom, the really small stuff, and we never knew what it was. We said it was degraded RNA. Turns out that these are microRNAs of great importance. Really interesting area of research. And finally, it turns out that there are quite a number of microRNAs in viral genomes. There are a number of ways to look at this. You have to do bioinformatic scans of the sequences and then do functional assays. There seem to be a lot in the herpes viruses. Many of these, again, aimed at antagonizing host functions. And these, remember, are viruses that go latent in you, as you will see. So it's likely that these microRNAs play roles in latency. The smaller DNA viruses, polyoma, even have a few. Adenoviruses have 2. These are large DNA viruses. They have a couple. And then retroviruses, it's a bit controversial whether they encode them or not. But in general, we think these microRNAs block apoptosis, that is turned on by the cell when it's infected. They help the virus escape immune responses, cell cycle arrest, which you will see happens when virus infects cells. And in the case of herpes viruses, probably promote latency. So if you're thinking of doing viral research, this is the area to go into. It's brand new and it's exploding.